Hello everyone, I am Biplab Bhattacharya and I welcome you all in this course Introduction to Sequence Stratigraphy. And in today's class, we are going to discuss about something different which is not directly linked with sequence stratigraphy, but this particular aspect is one of the very important aspect in sedimentology, particularly in recent times, and that has also created a special place in sequence stratigraphic interpretations because of its unique nature. So, today's topic is ichnology, ichnofabric and ichnofacies, and we will try to discuss about some basic aspects of this particular topic, and this discussion will continue into the next class as well. So, ichnology, it is not that common term and this is also not in the common purview of this particular course, but since nowadays ichnology has become very important in terms of interpretation of the paleoecological conditions, which are often controlled by various factors like autogenic and allogenic processes. So, the understanding of ichnology becomes very important many a time for interpretation of the sedimentary environments, for interpretation or identification of facies, and definitely when we are talking about sedimentary environment and facies, it goes into the purview of sequence stratigraphy. So, that is why I will try to give you a very brief idea about this particular aspect, ichnology, ichnofabric and ichnofacies. So, ichnology, this is the study of traces made by the organisms. So, this include the description, classification and interpretation of the trace marks of the organisms. So, this is the activity of the organisms which are preserved in the rock or within the sediments, but the organism is not present. So, when a sediment layer is getting deposited, at that time we know that organisms, they thrive on the sediments, they may thrive into the sediments for different life activities. The imprint of those life activities may often get preserved within the sedimentary record, within the sedimentary beds, and we call them as trace fossils. So, the organism which is responsible for making this is not present, is not preserved in the rock, but its activity is preserved. So, trace fossils generally reflect the activity of soft bodied organisms. Hard bodied organisms can also have imprints, but for hard bodied organisms we have a chance of getting their preservation in terms of body fossils, because hard part of the organisms may get preserved as body fossils. But for the soft bodied organisms, or maybe for the soft part of the organisms which are commonly not preserved in the rock record, we can get idea about them, about their activities, about their behavior through the study of trace fossils. In many environments, such soft bodied organisms represent the dominant component of the biomass. So, in paleontology, when we study the fossils, only few organisms are preserved. Say in one horizon we may get preservation of some bivalve, some gastropods, some cephalopods, and for them the hard part of their body are preserved. But maybe several worms were there, several annelids were there, and they do not get preserved in the rock record. Commonly in the siliciclastic rocks, they do not get preserved. So, those tiny small organisms which constitute a major part of the biomass are vanished. So, we can get an idea about those organisms and their life pattern, their life modes through the study of trace fossils. These activities of the organisms may get preserved in different mode, which we call as the structures. So, what are these structures? We can get four major types of structures in terms of activity of organisms. So, first one is the bioturbation, where organisms interact with the sediments. So, there we can get tracks, we can get trails, we can get burrows. So, what are tracks and trails? When some organism is moving on a particular substrate and it is leaving behind an impression of its movement due to some body part of that organism is in contact with the substrate. So, definitely the substrate should be soft, and on that soft substrate the organism is leaving behind some trails, some continuous or discontinuous marking due to its movement. Say for example, a dinosaur. is moving on a soft substrate and its footprints are getting preserved. In the rock record, we get those footprints preserved and we call them as dinosaur tracks. A gastropod may be moving on the seashore on a muddy substrate, and there it may leave behind a continuous marking. So, when the organism is in contact with the substrate for a continuous phase and making a continuous line, we call it as a trail. When the organism is making discontinuous marking, that means its body part is coming in contact with the substrate at certain intervals. For example, dinosaur, when a dinosaur is walking on the substrate, it is making its footprint at certain intervals. When we walk, human beings, when we walk, our footprints are preserved at certain intervals. So, these are tracks, human tracks, dinosaur tracks, and burrows are when organisms interact with the sediments and make different type of changes within the sediments. So, sometimes they may make a home within the sediments, they may churn the sediments, use the sediment for their food, they may use the sediment for moving from one part to the another, they can use the sediment for predatory reason. So, different reasons can be there, and due to those physiological activities, those organisms, they leave behind certain distinct features where the sediment, original sediment layers, they get little bit modified and the original layers are destroyed. Sometimes we call them as bioturbation, and this kind of structures are burrows. In the study of trace fossil, the study of ichnology dominantly deals with the bioturbations. But along with that, we can also get some biostratification where some organisms may help in development of or deposition of sedimentary rocks. So, for example, stromatolite. All of you know about stromatolite, and stromatolites are this kind of biostratification structure where some blue-green algae or cyanobacteria, they adhere the carbonate grains or phosphate grains and help in gradual building up of individual layers of the carbonate rock or the phosphate rock, and in that way we get columnar structure of stromatolites. Many times we can get microbial laminations within carbonate rocks or phosphate rocks. Also in siliciclastic rock we get biogenic mats, which also helps in development of different type of stratification within siliciclastic rocks. So, biostratification is a kind of bioaggradational structure which helps in the aggradation of the sediment layers. Apart from there, we can get some biodepositional structures like the fecal pellets, which are the products of bioerosion. Fecal pellets can also be generated by organisms during their feeding process, and they get deposited as layers within the sediments. So, in that way they form biodeposition, and also bioerosion, where organisms, they try to scavenge or they try to make hole within the hard rock or maybe within woods. That means those hard materials, within that they can make a boring, they can make a hole, and they can use that for different purposes. So, there definitely the hard rock portion is eroded, removed, and we get a new structure formed, which are basically bioerosional structure. Most of them comes under borings, some of them may be scrapings, scavenging, biting, etc. So, these are the broad framework which we deal in ichnology, but for this particular course we will focusing more into the sequence stratigraphic aspect, and from that aspect we will mostly consider this particular part, the bioturbation part. So, these are the activities of soft bodied organisms which commonly thrive during the time of sedimentation. So, syn-depositional or just post-depositional activities of the organisms are very important, and that we will deal in this particular discussion under the ichnology topic. In ichnology, these trace fossils, which actually reflect the behavior patterns of these organisms, have a very long temporal ranges. When I am saying this temporal range, that means we can get these different types of trace fossils across a very wide time range. I can take one example. Scolithos is a very common form which may be produced by some organisms, maybe a worm, maybe an annelid, even maybe some crustaceans, and they form whenever we get some high energy sand in the marine conditions, particularly in the shore area. So, that can be formed by organisms which are present in the Paleozoic. We get same kind of structure formed by organisms which are present in the Mesozoics, and also we can get them in the recent environment as well, because it is based on the activity of the organism in response to the surrounding conditions. So, this wide time range of the trace fossils helps us in the paleoecological comparisons of rocks of different ages. So, this is one example how trace fossils are identified or how trace fossils are used. So, we solely rely on the behavior pattern of the organisms, but the organism is not present. So, the behavior pattern of the organism which comes under this term called as ethology. So, let us take one example over here, say one trilobite, a trilobite resting within the sediments. So, at that time it will produce one kind of mark as we can see over here. Then it starts furrowing, so it will produce some other kind of structure. During that furrowing process, this kind of marking will be produced. When it will be emerging on the sediment surface, it will produce this kind of structures. When it will be walking on the sediments after emerging out, it will produce this kind of structures, and when it will be striding, it will be producing this kind of structure. So, the same organism on the same substrate producing different type of markings based on its different activities. And in case of ichnology, we identify these different markings as different trace fossils, irrespective of that these are made by the same organism or these are made by different organisms. So, when we get this kind of resting trace, we call them as Russophycus, this is the name of one trace fossil. We call them as Cruziana when we get this kind of furrowing marks. We can call them as Diplycnites when we get this kind of walking or striding marks on the soft sediments, and these may get preserved in the rock record. So, based on this ethology, we can classify the trace fossils into several groups or several types. So, we can get some of the organisms using the sediments for domicile purpose, for making their home. So, we call them as dwelling burrows, or in terms of ichnology, we call them as domicnia. Some of the organisms may use the sediments for feeding purpose, they search for food. So, we can get some feeding structure or photinychnia where you can get this kind of structures, and in that way we can get some grazing structures which is called as passychnia. We can get some walking structures which are called as rapychnia, resting structures called as cupychnia, escape structures called as fugichnia, like this. So, ichnia is the term or the suffix used to represent a particular ethological class or ethological type, and these ethological types actually indicates the different paleoecological conditions which are prevailing at the time of this burrowing. We can take one example over here again to understand this particular aspect. Say one organism is making a dwelling burrow, domicnia. So, where organism will try to make domicnia? As you can see from this drawing, this sketch over here, domicnia is a structure like this. So, organisms will try to come down from the substrate to come over here. So, definitely if the organisms are lying on the substrate, they will not make a domicnia, they will not make a home over there, but they will try to search food. So, when they are searching for food, they will try to cover up as much as area possible on that particular surface, because they can scavenge or extract the food as many as possible depending on their amount of area coverage. So, a photinychnia will be on the surface dominantly, or it can go down little bit if the organism is searching for food in a particular layer. But when an organism is resting on another surface and that surface is bit disturbed, say for example there may be some current is going on or maybe some high energy waves are going on, so definitely the organism will not like to thrive over here. It will try to go down, or it will try to find out some area where it is more stable, undisturbed. So, if the organism does not have the choice to move into other place, then what will do? It will try to go down the sediment, making a hole like this, a vertical or inclined hole, going down up to certain extent, and then it will try to reside over here, stay over here. And commonly this kind of organisms, they collect their food from the suspension, we call them as suspension feeder. That means whatever elements are available in that water body in suspension, they collect that food using maybe some tentacles or something. Whereas these organisms which are making photinychnia, making some trails or some markings on the surface, searching for food on the surface, they are actually looking for the sediments and the food which is already deposited on the surface. So, these are called as deposit feeders. So, based on this nature of deposit feeding or suspension feeding, organisms can make these different types of structures. So, one will be dwelling structure, another will be feeding structure. Now, if in one place the amount of nutrient is very high and that condition is very much characteristic for a particular type of organisms in terms of energy condition, in terms of substrate condition, in terms of the overall availability of nutrients, then organisms will try to scavenge that entire area in a very closed spaced manner. So, in such cases we get this kind of structures, grazing, which we call as pasicnia. So, definitely this indicates that the organisms having high population were only concentrated on the surface and they were grazing for some food which were available on that particular substrate in high amount. So, organisms were trying to scavenge all those foods within a small area. Whereas, if an organism has to move from one place to another and they are not looking for food, or maybe they are looking for food but that is available in another place, so they will not go for this kind of grazing at that time, but they will just move from one place to another. So, we will not get the grazing trace over there, but we will get this kind of walking trace or rapicnia. So, each and every type of behavior is actually in response to certain changes or certain conditions available in the sedimentary environment. These minor organisms, these tiny small organisms, particularly the invertebrates, they are very much sensitive to minor changes in these conditions. So, as soon as there is a change in the ecological parameters, their behavior will change, and when those behaviors changes and it gets preserved in the sediments and then gets preserved in the rock, we can get idea about those paleoecological changes through the study of these trace fossils. So, in ichnology we follow certain basic principles. So, the first basic principle that we follow is that an individual trace fossil may be the product of one organism, like we have seen the trilobite example, and in that case it becomes easier to interpret, or sometimes a trace fossil may be the product of two or more different organisms. Commonly we call them as the composite structures, and it becomes more difficult to interpret. Many times we can get say one gastropod trail and then it has been overprinted by a bivalve trail. So, that pattern of that particular trail becomes very complicated. Similarly, an individual organism may generate different structures corresponding to different behavior in similar substrate. Again, I will ask you to refer back to that example of trilobite trace, where that single trilobite was making different type of traces by different activities on the same substrate, or it can produce identical behavior in different substrates. So, the same organism can produce different type of traces when the substrate condition changes, because as the substrate condition changes, its behavior will change. It may change its feeding pattern, it may change its locomotory pattern, and because of that the type of ichnoforms, the type of the trace fossil will change. At the same time, identical structures may be generated by different organisms with similar behavior. As I was giving you the example of Scolithos. So, Scolithos can be produced by different organisms like annelids, like worms. So, these different organisms in different conditions can produce similar vertical burrow structure, and depending on the morphology of that burrow, we call them as Scolithos burrow. So, these organisms, they are very much sensitive to several paleoecological factors, parameters. For example, they are very much sensitive to energy condition which is prevailing in that particular regime, in that particular depositional environment, or to be more precisely in that particular part of the depositional environment. They are very much sensitive to substrate condition, what type of sediments are getting deposited, what type of sediments are coming into the system, and other ecological parameters such as salinity condition, oxygen levels, supply of oxygen during sedimentation near to the sediment water interface, within the water, within the sediment, rate of sedimentation, luminosity within the water body, because as the luminosity will decrease, the photosynthetic organisms will decrease, and that will decrease the availability of food for many organisms. Similarly, temperature, because as the temperature will go down, it is not supportive for many organisms to thrive. Similarly, if the temperature goes very high, in that case also many organisms cannot thrive. So, temperature is also a very important factor, and the abundance and type of nutrients, which guides that what kind of feeding activity, what kind of life activity will be used by the organisms. So, when all these factors, all such ecological factors are present within a particular sedimentary environment, and that is guiding the nature of biomass and their nature of activities within that particular sedimentary environment. So, definitely minor changes in any of these factors or minor changes in multiple factors out of these ecological parameters will definitely lead to change in the behavior pattern of the organisms. So, trace fossils indicate those changes. If we can study the trace fossils in proper systematic way through this ecological study, we can very easily understand and interpret about those minor changes in the paleoecological condition. And in rock record, this becomes very important, because say some rock which has formed maybe 200 million years back, maybe during the Paleozoic, maybe during the Mesozoic. So, nobody has seen what was the conditions over there at that time. So, studying this kind of trace fossils present within those rocks can give us guideline about those paleoecological parameters 200 million years back. That is why ichnology has become a very important parameter nowadays in sedimentological study. The behavior patterns of these organisms depend on the ecological conditions which relate to the particular depositional environment. So, we commonly use or go through two different modes of studying. One is called as ichnofabric and the second one is called as ichnofacies. So, in today's class, I will try to give you idea about the ichnofabric, the concept of ichnofabric and what we commonly do in ichnofabric. And in the next class, we will discuss about the ichnofacies part. So, ichnofabric, it is a new term which commonly indicates all the aspects of the texture and internal structure of a substrate that result from bioturbation and bioerosion at all scales. So, originally a sediment layer was deposited. Now, some organisms are interacting with that sediment layer, or maybe when the sediment layer is getting deposited, at that time some organisms are interacting with the sediment layer. And as a result of that interaction, what is happening? The original fabric of the sediment layer is getting disturbed or maybe to some extent getting destroyed, modified. And when that layer is completely deposited and that organic activity is stopped, then what we are getting is a new fabric, a modified fabric from the original one, original sedimentary fabric. This new fabric is produced by the organic activities, the organism's activities, and we call them as ichnofabric. So, ichnofabric is basically this new aspect, new appearance of the rock or that sediment bed due to these organic activities and the trace fossils which have formed within that particular rock body. So, when we go through this ichnofabric concept, it deals with the recognition of the characteristic ways in which the texture and fabric of a sediment has been affected by organisms' activity. So, to study this, we have different tools and we apply those tools systematically one after another to identify different parameters which help us to understand what is the ichnofabric, and using that ichnofabric we try to interpret about the paleoecological conditions. So, this is a very traditional diagram used in ichnofabric analysis where we try to define different parameters which contribute to the ichnofabric. So, this part is the conceptual part. These are the different parameters. For example, the type of the biota. So, it may be deposit feeding, it may be suspension feeding, it may be farming. What is the ethology or the behavioral pattern of the organisms? What is the nutrient supply in that particular depositional regime? What is the oxygen supply? Because depending on oxygen supply, the nature of behavior of the organisms will vary, the type of organisms will vary. The state of the substrate, it may be a loose substrate, that means it is a soft condition, sediments are just deposited, they are not consolidated at all, or it can be a firm substrate where the sediments are partly consolidated. or it may be completely consolidated, we call it as a hard ground or hard substrate. So, whether the ichnoforms, whether the bioturbation is occurring in loose or soft ground condition, or they are forming in a firm ground condition, or they are forming in hard ground conditions. What is the sedimentation rate? Because as the sedimentation rate becomes faster, so within a given time more sediments are coming into the system, and also the energy condition possibly is little high. So, in such condition the density of the organisms will be little bit less, because by the time organisms are burrowing, more sediments are getting deposited. Whereas, if the sedimentation rate is very low, then organisms get lot of time for scavenging, for doing several activities. So, in such cases we get more bioturbations. So, sedimentation rate is one important aspect which can be identified and which also help us to determine about the ecological conditions. Colonization window is another important parameter which develops maybe in terms of certain time intervals. Certain conditions are prevailing which is helpful for the growth or blooming of certain organisms. So, during that particular time period we are getting a colonization of one particular organism or a different type of organisms within a particular depositional environment. When that window is over, then there is no colonization and we do not get any kind of bioturbation. So, this particular concept is very important in identification of some of the important surfaces in sequence stratigraphy. We will discuss about this later on in much detail. Another important parameter or concept associated with ichnofabric is tiring and ordering, where many times some of the organisms they prefer doing their activities on the surface or very near to the surface, whereas some organisms may try to go down, and in one given time we can get a structure like this, a rock body having this kind of structure where different types of organic activities are preserved together, but it is not necessary that all of them were formed at the same time. They have formed in different times in different orders. So, some of them have formed when the sediment layer was over here, when the sediment layer was over here, whereas some of the structures may have come down from this particular layer much later, but they have come down to this layer and they are now overprinting the earlier forms which were present at this bottom layer. Understanding that which one is the earlier, which one is the later, and understanding that from which layer and which condition they have formed, how much depth they are penetrating, that comes under this particular concept, tiring and ordering. And finally, the parameter diagenesis, which often helps in the preservation of certain ichnoforms, or sometimes it destroys some of the ichno patterns, some of the ichno texture. So, sometimes we get some kind of enhancement due to compaction, the shape of the ichnoforms may get changed. So, we have to take care of these diagenetic changes when we study the ichnoforms from the rocks. So, these are the different parameters which contribute in the formation of ichnofabric. But when we are looking into a rock and we are trying to understand the ichnofabric, we have to represent them. For that representation purpose, we characterize the ichnofabric using several parameters. So, number one is the bioturbation index, which indicates that how much bioturbation has occurred within a particular sedimentary unit and how much of the original sediment layers are preserved or destroyed. So, definitely it is associated with the number of ichnoforms, the density and diversity of the ichnoforms present in that particular rock body, and how much the original layers are destroyed. So, if the number of the ichnoforms is less, the chances are there that the original laminae is almost preserved. So, we designate them as a low bioturbation index, whereas there may be conditions where the density of the ichnoforms are so high that the original laminations are nearly destroyed or completely destroyed. So, in such cases the bioturbation index will be very high. So, it will be around 4, 5 or 6. So, there is a designated scale available which we refer in terms of how much of this bioturbation is present and how much of the original lamination is destroyed. Similarly, we try to represent in terms of ichnofabric constituent diagram, where we try to represent in terms of relative proportions of different events and their resultant products. say for example deposition of some layers initially, which are then obscured by some other activities, some organisms, which is further obscured by other activities of organisms, like this, and how much percentage it is covering within that particular unit. So, that gives us idea about the different components which are present in constituting that particular ichnofabric. Also, we take care of the diversity, burrow size and the frequency of different ichnoforms, and using all of these we try to make tier diagrams where we try to represent what are the sequence of events of formation of these different ichnoforms within that particular sediment layer. So, these are the different ways of characterizing the ichnofabric. So, definitely we do not have much scope of going into the detail of this particular concept. So, what are the conditions that we can interpret by studying this ichnofabric analysis? So, we can interpret about the energy condition, whether the wave energy condition, current energy condition, whether it is a high energy condition, low energy condition. We can interpret about the temperature condition in that particular ambience. We can interpret about the salinity and quantity of available oxygen in that particular depositional regime near to the sediment water interface. We can also interpret about the light penetration, as I told you earlier, luminosity, because light penetration helps in growth of certain organisms, helps in the growth of certain nutrients as well. How much is the nutrient supply is a very important parameter, because nutrient supply controls the nature of organisms and their density. Also, the competition for ecospace and resources. Say you have in some areas lot of organisms, so there will be competition for the food, and that will definitely lead to decrease in number of organisms, but initially there will be lot of perturbations. So, the competition for ecospace and the resources is another prime factor which determines the nature of behavior of the organisms. Sedimentation rate, we have discussed about that already, and definitely the nature of the substrate, whether it is a soft ground condition, whether it is a firm ground condition, whether it is a hard ground condition. When it is a soft ground condition, then within that soft ground condition or soft substrate condition, what is the grain size available, whether it is a sand dominated, mud dominated, clay dominated, because that will control what kind of organisms can thrive over there. That is related to the energy condition, that will control how much nutrients is available over there and what are the properties of those sediments. So, looking at the ichnofabric, we can interpret about all these parameters. So, these are the references that you can go through for further reading, for understanding about this particular concept. However, we will continue this discussion in the next class, and there we will discuss about another aspect of ichnology, that is the ichnofasis concept. So, thank you everybody.